Restriction enzymes provide other opportunities. If we combine digestion with restriction enzymes with DNA ligase, key in DNA replication if you will recall, there are new opportunities that emerge. Restriction enzymes recognize specific sequences and generate overlapping ends, and that provides a special opportunity because the sticky ends of restriction endonuclease digestion can interact to form a semi-stable complex. And that complex can be recognized by DNA ligase to reseal the DNA to its original sequence. But remember, ligase is not sequence specific. It will reseal any nick in a DNA sequence. And this feature of ligase makes it possible to recombine DNA from different sources as long as the sticky ends match to create a nick. DNA that has been generated by restriction enzyme digestion and then inserted into another DNA using DNA ligase is termed cloned or recombinant DNA. And the sealed ends retain the original restriction endonuclease target site so that treatment with that enzyme will recover the inserted DNA. Now, plasmids are common carriers for cloned DNA. These small, usually circular DNAs are separate from the chromosomal DNA in bacteria with independent replication processes. They occur naturally in bacteria and occasionally in eukaryotic organisms. Easy to isolate, plasmids are amenable to insertions of DNA, creating plasmid combined with inserted DNA, which is called recombinant DNA. Because plasmid DNAs replicate within the bacterial cells, growing bacteria generates large amounts of plasmid DNA and expression of any proteins that are encoded by the recombinant plasmid. This method allows isolation of significant amounts of proteins for which DNA coding information is available. But the protein itself may be produced naturally only in small amounts. So this approach is one of the key methods of modern biological sciences where proteins even produced in small amounts in the cell can be produced in large amounts and characterized.